So for our chapter 13, our second set of boxes is DNA, the genetic material. Our previous set were on the experiments leading up to the discovery that DNA was the molecule of inheritance. Okay, so when we look at nucleic acids can be both DNA and RNA, and their job is to store and transmit hereditary information. But the key here is that their monomers are nucleotides. So when we look at DNA, the double helix, it's a long chain of nucleotides over and over and over again. In us, every single cell in our body has 3 billion base pairs. Now what that means is a base pair is when an A pairs with T and a C pairs with G. We have 3 billion of those in every single cell in our body. And so when we look at what we mean by A, T, C, and G, we're talking about nucleotides. So right here is one nucleotide, and a nucleotide is made of a nitrogen base, a phosphate, and a sugar. We saw in Messelson and Stahl that we have our phosphate, a 5-carbon sugar, and a nitrogen base. Okay. So DNA is made out of a nucleotide. The nucleotide's 3 parts are phosphate, 5-carbon sugar, and a nitrogen base. And it's those bases that change but there's 4 different kinds of nitrogen bases. A, T, G, and C, thymine, cytosine, adenine, and guanine. So in box 1, please draw a nucleotide and label it. Now, we have 2 different categories of nucleotides. We can have what's called purines, which you see here, it's a double ring structure. And we have pyrimidines, which is a single ring structure. Now, purines are A and G, and pyrimidines, the way I remember that they're C and T is that cytosine and thymine both have Y's in it. So when I look at the 2 categories of nucleotides, I see here that my purines have a double ring structure, and my pyrimidines have a single ring structure. Now, when I look at the significance, we only can have purine-pyrimidine pairs that fit inside of our double helix. If you try and fit 2 purines together, it's too crammed and there's not enough space. If you fit 2 pyrimidines next to each other, it's actually too much space. That's too large of a gap. But when you put a pyrimidine and a purine next to each other, they fit just right. So we see, when we look at DNA, that we have a purine and a pyrimidine. So here's my purine, here's my pyrimidine. They pair together. Okay. So what are purines, what are pyrimidines, and how do they pair up? All right, so when we look at building a polymer, a nucleic acid polymer, we start with our building block, which is a nucleotide. Now, when we go to attach a second nucleotide and build it in a chain, we have our phosphodiester bond. So the phosphodiester bond is right here, connecting the phosphate to the sugar. So there's your next nucleotide, it attaches right here. The next nucleotide, you have your phosphodiester bonds. Phosphodiester. Again, so you can see how DNA is going to be built in a repeating chain of monomers. The monomers are nucleotides, and they're connected to each other with phosphodiester bonds. What we've just built here, this part is called the sugar-phosphate backbone. Okay, so when we look at putting 2 of those together, here's the sugar-phosphate backbone and the sugar-phosphate backbone. What we see is that the hydrogen bonds are what hold the base pairs together. Between A and T, there's 2 hydrogen bonds. Between C and G, there's 3 hydrogen bonds. Okay. Ooh, and with DNA, if we know one side, so let's say we know this side, and we have no idea what the second strand is, we can figure out if we have a T, we know that it pairs with an A. If we have a G, we know it pairs with a C. And this is what's said to be complementary. If I know one side of the DNA, I can figure out the other side. DNA is said to be complementary if you have one strand. So let's say I know one side is this. Then I could easily find the other side because of base pairing rules. Base pairing rules lets me know that if I know one side of my DNA, I can figure out the other side because it's said to be complementary. All right, so what does it mean that DNA is complementary? Goes in your box 3. All right, another thing we're looking at in this unit is what's called the central dogma of biology. This is the flow of genetic information in a cell. Now that might mean how you get from one cell to 2 daughter cells through mitosis, but it could also mean how you get from DNA to RNA to a protein. So in chapter 13, we're looking at replication, and in chapter 14, we're looking at transcription and translation. But all of these, we're talking about the flow of genetic information. So we're focusing on replication in this part. Okay, so DNA is a review. We have hydrogen bonds between the base pairs, and we also have phosphodiester bonds in the sugar phosphate backbone. Now you'll notice these 3s and 5s, which right now don't mean anything, but as you review before your test, you're going to want to pay careful attention to the 3s and the 5s. So when we look at nucleotides and we count the carbons, we have a carbon number 3 here, carbon number 5 here. It's the carbon number 3, we call it 3 prime, that's what this little area means. And here's 5 prime. We talk about DNA's direction in terms of 5 prime to 3 prime. So when we are building a DNA, what kind of bond is that? Good, it's a phosphodiester bond. So if we see here's 2 nucleotides, I can see here's my 5 prime end, there's my 3 prime end. We want to look at the last trailing carbon. And at the bottom here it's a 3, and at the top here it's a 5. So what I say is this is the 5 prime end, and this is the 3 prime end. Oops. Okay, so when I look at my DNA, what I see is that here's a 5 prime end. If I notice and look careful, here's one nucleotide. Look at this one. Look at the T, how it's upside down. And this one, same thing with the C is right side up. Oh my, this one's upside down. Oops, I forgot the phosphate. So when we look at our DNA, what we actually see is that one strand goes from 5 to 3. Here's my 5 prime end, my 3 prime end. And on this side, it's the opposite, 3 prime to 5 prime. When we say this, so this DNA is going from 5 to 3, and this one's going from 5 to 3. They go in opposite directions, so they're said to be anti-parallel. All right, they run in opposite directions. Complementary strands run in opposite directions. Okay, so if I wanted to replicate DNA, here's my parent strand in blue. This is Messelson installed when they used N15, by the way. And then as it unzips, we have the original here in blue. And then we have the new parent strand in orange. So the blue actually acts as a template or a new strand for the blue. So we have half, the new strand of DNA is half the parent. So if I were to have just this right here as my DNA, I could see half is the parent and half is new nucleotides. Oh, oops, dang. Okay, so when I look at replication, now the first step in replication is we need to unwind the DNA. So here we have an enzyme called DNA helicase. Now DNA helicase moves and unzips and breaks those hydrogen bonds. And then we're going to use single-stranded binding proteins to hold open and stabilize that helicase. And then here we have what's called the replication fork. So this is like a fork, and it's going to open this way, and it's going to keep on unwinding. Okay, so when we look at how we build a new complementary strand, and as we said before, if I know one side, I can figure out the other. So this would be base pairing, but how we do this is we're going to need to use an enzyme. So the enzyme we're going to use is DNA polymerase 3. So DNA polymerase 3 is going to add nucleotides, and build it. You'll notice that these nucleotides are upside down. If I see here's my 5', here's my 3', here's my 5', here's my 3'. So when I look at these strands, though, let's watch that again. So as I pay attention, I want you to look at the nucleotides. So right here's adding to the 3, adding to the 3, adding to the 3. The new nucleotides are built adding to that 3' carbon. But now, when we think about this, what are we missing? How do nucleotides form this phosphodiester bond without an investment or input of energy? If we're building a bond, that's got to be endergonic, right? Okay. So first, though, let's go ahead and write down what is the job of the enzyme DNA polymerase 3. All right. So now that energy, where does it come from? Well, what does this look like? This looks like ATP. So ATP truthfully is just a modified nucleotide. And we've already learned when you break off a phosphate to get ADP, you release energy. Well, if we break off 2 phosphates, that also releases energy. And now we have what's called a nucleotide. Awesome. So when we look at this, the nucleosides with 3 phosphates come with their own energy, and they leave behind a nucleotide. So when we look at our GTP, it brings its own energy. You have TTP. You also have CTP. All right. So when we look at these molecules, we call them nucleosides. Nucleosides have 3 phosphates. They bring their own energy source to bond and connect to the next phosphate or the next nucleotide. So in your box, oh no, just kidding, just check yourself, the difference between a nucleoside and a nucleotide. Okay, so let's go ahead and see how this happens. So when we add bases, we can only add on, or DNA polymerase 3 can only add on to the 3' end, but it does need a starter nucleotide to bond to. And it only grows in the 5' to 3' direction. So here's DNA polymerase 3, going to break off those 2 phosphates, releasing energy, and that energy is going to be used to build the phosphodiester bond. Here's your next nucleoside with DNA polymerase 3 that's going to break off 2 phosphates, releasing energy, and we're going to build that phosphodiester bond. Now the TTP nucleoside comes in, breaks off 2 phosphates. We're going to take the energy and build this phosphodiester bond all the way through to build that complementary strand. Now, how do we build that complementary strand? We build it based on base pairing rules. We've built phosphodiester bonds in the sugar phosphate backbone, and there's hydrogen bonds here holding the base pairs together. Now, what we've also noticed is here's the 5 and here's the 3. So what we have is we've actually built this DNA from 5 to 3. That's the direction we built it in. We say this DNA molecule was built in the 5' to 3' direction. So when I look here again, DNA polymerase comes, breaks the energy, builds the bonds. You can see now how we're adding it to the 3' end. The 3' end is where we're adding the next nucleoside because the energy is going to be right here when we break off those 2 phosphates to build that phosphodiester bond. So our DNA is built in the 5' to 3' direction. So if I separate the 2 strands though, and I can build this one, I'm going to identify my 5s and my 3s. Now when I build it, cool, I can build it continuously, attaching to the 3' end. And I can see here that this was built in one continuous motion. Whereas the other side, if I go to build this in the 3 to 5, it doesn't work because the energy is down here. So how am I going to connect this? I can't. And this is why we build it in the 5' to 3' direction. And we'll actually build it in fragments on this other side. And then right here, we're missing a phosphodiester bond.